I want to make sure we give enough time and attention to our audience Q&A. And so this first question, Mitch, I'd like to put to you, which is, in today's climate, when there are more options for CDMO in the gene therapy space, would you recommend early stage organization to leverage their use to expedite their clinical trial manufacturing needs versus building an in-house facility?
So I think it comes back to the number of programs you have. If you are, and there's a critical mass number, probably somewhere between 4 and 6, depending on where those programs are. If they're relatively close together, then the internal capabilities offer a lot of flexibility and control, reacting to changes in timelines and developments in your non-clinical data and so on. If you have sort of below that critical mass, then the CDMO market has definitely evolved over the last, I'll say, 2 to 3 years, where I think there's some really good opportunities and options out there. So it's certainly a viable opportunity.
Okay. Will, I'll address this question to you, but Mitch, please feel free to provide a response if you'd like. Was the speed of implementing process changes a factor in build versus buy? Build versus contract.
Absolutely. Yes, absolutely. For sure. It's much easier to introduce a process change in-house than to work through contract negotiations and make it at a CMO.
Yeah,
sure.
Absolutely. 100% agree.
Okay. So Mitch, we'll just continue with you here. How do you work out the initial budget requirement to build your own facility? We did talk budget at the top end of our call, but perhaps a little bit more on this. What is the starting point of the process to define the correct cash upfront, and what are the key points of consideration?
Yeah, so for us, as I mentioned, we didn't really have a process to build to. So the head of development and myself and the construction guy sat down and we just sort of mapped out a plan to get us started. And we really focused on what is absolutely required and what is a nice to have. And again, we were pre-Series A, so money was definitely a big driver in keeping that number as low as possible. We really focused on flexibility. So larger, I think Will mentioned earlier, ballroom design. Keeping the overall building cost down. Pretty unusual deployment, but our spread was about 65% equipment and 35% building, which is a pretty good number. I'm pretty proud of that, honestly. But what that did for us is allow us to get going on the building and then continue to evolve the process design so we could buy the correct equipment. And in a few cases, we needed additional pieces, but everything being portable and single-use technology for us was relatively easy to adapt to some of those process changes we mentioned a minute ago.
Sure. Okay, Will, would you like to add anything to that regarding
I completely
budget
agree.
considerations?
Yeah, completely agree.
Okay. All right. Let's talk about mitigating risk. So how would you or do you mitigate the risk in manufacturing of high-dose cell and gene therapy products? Just your overall take on that question. Mitch, can you? Yeah.
Well, go ahead, Mitch.
I'm not sure which risk I'm focusing on, but I would say, the early manufacturability assessments are really important to ensure we achieve, highly pure product, removing all the contaminants. From a cost of goods perspective, you just have to make sure you consider the anticipated dose and how you would factor that back into your capacity analysis for supply. I hope that answered the question. I'm not sure I fully understood it, but
I think it's a good answer. We're good. Let's move on to another one. We'll start with you. Can you share some of the challenges encountered during comparability demonstration during cell and gene therapy manufacturing?
Comparability with what?
That is a good question. Comparability just perhaps in general, but this is not really outlined here, and that's okay. We can perhaps get back to this person later after the fact, and we can get a better understanding of what this person is trying to ask. That's no problem. What pods do you recommend?
What?
Or perhaps some the modular designs. Are there any that you have used or recommend, or just know of any in the industry that are helpful when using prefab modular units or modular design?
We have not used any prefab modules at this point. And I'm not quite sure about the breadth of the industry out there, of the suppliers, so I wouldn't be able to answer that question theoretically from a who would I go with perspective. Lastly, it's about the user requirements. In other words, what do you really need that pod to do, and then get bids from the market.
Sure.
Yeah, I would, I think that's absolutely right. Get your user requirements first, and then, work with an engineering firm to assess the marketplace. There's a lot of providers now. I say a lot. I know of at least 4 or 5, and we've evaluated several of them. But the engineering firms usually know, how to match your user requirements with the right product. We can help you work through a bidding process.
Gotcha. Okay. All right. I think that actually gets us through many of the questions that our audience has shared with us to date. I'm just trying to get back to my information here. And finally, just from a theoretical, best moment during the development to build an in-house plant, just to kind of put a period at the end of this sentence, any final thoughts for our audience who are considering building their own facility, or in the middle of building their own facility? What are kind of your words of advice at this point before we sign off? I'll start with you.
Oh, wow. Okay. Yeah, keep the process safety and risk associated with actual processing in the forefront of your mind. There are many, at least from a cell therapy perspective, many pitfalls on the way, from leaky bags all the way through, particles and final product and all that kind of stuff, right? So the risk to patient was for us and is for us and will be for us always like the number one driver, in design of the facility, as well as the qualification part, which equipment to use, which clean room technology to use, all that kind of stuff is so essential to get right off the bat. You can't go back and change your design in the aftermath and then while you are supposedly commercially manufactured.
Sure. Okay, thanks. And Mitch, final thoughts for the audience. Yeah,
I think hitting the critical mass and making that decision to jump all in. When you jump in. Don't. This is not for the faint of heart, right? Building this is. It requires a strong team. There's been a lot of clinical holds for people who didn't go in with the right amount of focus and commitment to it. So if you're going to go in-house, it's a great experience, and you should keep in mind or keep in your line of sight the benefits that we've outlined: knowledge retention, speed, flexibility, quality. If you keep those at the forefront of all your decision making, and you are committed to it, I think you'll end up in a really great place and be really happy with the results.
Good. Okay. All right, everyone, that's it for us today on this Cell and Gene Live event featuring our subject matter experts, Kite Pharma's Will Junker and Kriya Therapeutics' Mitch Lauer, and of course, our partner Integris. I'd like to thank Mitch and Will sincerely for their time and insight with us today, and thank you to our audience for your time and attention. I'm Erin Harris. Visit us at cellandgene.com for the on-demand version of this presentation, and we will see you next time.
Thank you.
Thank you.